Hello friends, good morning all of you. Here I am Gadekar Sachin Pandit. Today we are discuss ninth chapter terpenoids and alkaloids. Okay, so in this chapter we discuss first introduction, then classification and structural elucidation. Okay, so first of all, last lecture yesterday we are discuss combined UV, IR and PMR data used for structure determination. UV provides the information about conjugated double bond. Okay, so IR, it provides a presence of functional group. Okay, so suppose the IR absorption at 1750 centimeter inverse, it means it due to the presence of maybe carbonyl group, presence of aldehyde group or ketonic group. Okay, so these valuable information due to the presence of carbonyl absorption. Similarly, other groups are suggest other functional group. Then PMR. PMR suggests the presence of environment of proton. Okay, type of protons. These all information helps to determine the structure of compound. They are discussed in last lecture. So today we are discuss terpenoids and alkaloids. Okay, first introduction. Content, Contents definition of terpenoids, then discuss a property of terpenoids, isolation of terpenoids, classification, isoprene rule, isoprene unit, general method of structural elucidation, and then structure of terpenoids. And lastly, we discuss a citral molecule. First, definition of terpenoid. Terpenoids are naturally occurring hydrocarbons of plant origin. Of the general formula of terpenoids is C5H8 nth, means these C5H8 unit number of units are present in terpenoid molecule. Okay, so the terpenoid units CH8 C5H8 unit, it called isoprene unit. Okay, so this C5H8 is the 2-methyl 1,3-butadiene. There are two types of terpenoids: simple terpenoids and complex terpenoid. Okay, so simple terpenoids occurs in steam volatile essential oil and obtained from sap and tissue of certain plant and trees. Okay, sap is the secretory material of plant tissue. And then complex of terpenoids are not steam volatile and obtained from gum and resin of plant. Okay, so this is the terms of essential oil terpenoids oil. So example of terpenoid oil, geranol, it's present in rose. Okay, so this geranol present in rose essence. Okay, so second is the lemon. Limonene is also present in lemon. Okay, so this is the example of terpenoids. Xenol, these are present in clove. Okay, so then citral also present in terpenol. This is the example of terpenoids. Then physical property of terpenoids. Okay, terpenoids are the colorless liquid. Okay, they are colorless, soluble in organic solvent and insoluble in water. Solubility of terpenoids is in organic solvent, not water. Most of the terpenoids are optically active, means chiral, and volatile in nature, means its boiling point is very low. Boiling point near about 150 to 180 degrees centigrade of terpenoids. It's a physical property of terpenoids. Then second is the chemical property of terpenoids. Terpenoids are unsaturated compound. Okay, so use a isoprene unit. Okay, C5H8. It contains double bond. It means they are unsaturated. They undergo addition, reaction with hydrogen, halogen, halogen acids from addition product. Means double bond reacts with halogen to give addition product. They undergo polymerization and dehydration in the ring. Polymerization means one unit connects to another unit, means polymerize. Polymerization and dehydrogenation in the ring. Okay. The on terminal decomposition, terpenoids gives isoprene as one of the product. Okay, so next is the isolation of terpenoid. How to isolate? Okay, from plant. Okay, isolation of essential oil from plant part. Okay, so there are four methods for isolation. First is the expression method. Next is the steam distillation. And third is the solvent extraction. And fourth, adsorption in purified fats. Okay, so this is the four method for isolation of essential oil from plant part. Next is the separation of terpenoid. Okay, isolation of terpenoid, there are two most part. First is the isolation of essential oil and second is the separation of terpenoids. Isolation in which four part and then separation of terpenoid, it also three step. First is the fractional distillation. Then second is the fractional crystallization. And third is the chromatographic technique used such as TLC, GLC, HPLC and column chromatography. Out of these, column chromatography is mostly used because TLC, GLC, HPLC is most costly. Okay, more costly. So separation of terpenoids are three steps: fractional distillation, fractional crystallization and separation for using chromatographic, column chromatography. Okay, next, isolation of essential oil. First method, expression method. Okay, so in this expression methods, the plant material is crushed. First, plant material or leaves crush and then juice obtained is filter. Okay, so first plant leaves or plant material is crushed and the juice is obtained. Okay, so this juice filter, then this juice centrifuge. Half of the essential oil is obtained in the centrifuge. Then this solid residue is used for isolation of inferior oil by distillation. Okay, this is the diagrammatically representation of expression of method. Okay, first plant leaves crush, then they transfer in blending and then press pressure is applied to forms a juice. Okay, so freshly preserved juice they transfer and extract. Okay, juice which is get centrifuged and purify. Okay, so this is the expression method for extraction of juice. Next method is the distillation method. Okay, so in this figure, distillation used for preparation of isoprene essential oil, terpenoid. Next is the solvent extraction. In this solvent extraction, solvents like hexane and ethanol is used to isolate essential oil. Solvent hexane is the non-polar and ethanol is used. Okay, there are two types of solvents are used. Hexane and ethanol is used for isolate essential oil. Okay, so it is used for the plant part have low amount of essential oil, and then plant materials are treated with solvent and it produce a waxy aromatic compound is called concrete. Okay, so base these waxy material aromatic compound is called concrete. Okay, so base material, and this then it mix with the alcohol, the oil particles are released, and then these released oil particles press through condenser and then it separate out. So this oil is used in perfume industry as well as aromatherapy purpose, cosmetic. Okay, so these oil used for perfume industry, cosmetic, beauty, soap, etc. aromatherapy purpose. So this is the methods of extraction of terpenoids. Fourth method is the influorage. Okay, so this fat is warm to 50 degrees centigrade on glass plate. Okay, then this warm fat is covered with flour plate and it kept for the several days until it saturated with essential oil. Then the old petals are replaced by fresh plates and it repeated process. Okay, influence. Okay, after removing the plate, the first fat is treated with ethanol and then these oil present in the fat are dissolved in ethanol. Okay, so these ethanol distillate in the fractionally distill under reduced pressure to remove the solvent. Okay, so this is the method for influence. So rose petals influence for separation. Okay, so this is the methods. If any question, then ask, comment. Thank you for watching this video. Have a nice day. Next lecture we are discuss classification of terpenoids. Okay, thank you. Have a nice day.